Hello, my name is Molly Shook, and today I'll be presenting an ecotoxicological assessment of DDT. DDT, or dichlorodiphenyl trichloroethane, is a historical pesticide with very big environmental implications. It was first synthesized in the late 1800s by a chemist named Othmar Zeidler, and later on, a Swiss chemist named Paul Mueller discovered its insecticidal properties. By the 1930s, DDT was being very widely and successfully used to combat malaria and other insect borne illnesses. It was also highly successful in enhancing agricultural productivity during that time. By the 1950s, the world was beginning to realize DDT had some less favorable consequences. And in 1958, the U.S. Department of Agriculture initiated the National Organophosphate Program in an attempt to eliminate the use of DDT. Despite the concerns of environmental persistence and effects on non target species, many developing countries still manufacture and use DDT to combat diseases like malaria. That's why it's important to assess the risk and consequences of DDT and promote alternative pest control strategies that don't compromise ecological integrity. So let's take a closer look at the chemical structure of DDT to better understand why it's so persistent in the environment and how it's caused such a lasting impact. First, it's a colorless crystalline chemical represented by the chemical formula that you see on the slide here. Its legacy is significant. As I mentioned before, it was among the first pesticides to be broadly used for agriculture, pest management, and malaria control. It was rapidly adopted as the solution to a pressing economic and public health issue before the consequences were fully understood. And due to its resistance to degradation, we continue to deal with those impacts today. It's now known that DDT is toxic to many aquatic organisms, and one journal linked it to endocrine disruption and potential carcinogenic effects. The journal also reported LC50s to be as low as 1.5 microgram per liter for some aquatic species. DDT has also been shown to weaken avian eggshells, which severely impacts reproduction rates. And this compound is a key example of bioaccumulation and biomagnification, which further highlights the need for stringent testing and oversight. Its properties allow it to accumulate in the tissues over time, but we'll dive much further into that later on. Before DDT was banned in the US, it entered the air, water, and soil through its use as an insecticide. Large quantities were released during pest control sprays, and today it remains at many waste sites, continuing to pollute the environment. While waste sites pose a significant threat of entry, DDT can also enter the environment from industrial sources and ingestion. In humans, contaminated fish consumption is a big issue, but inhalation and skin contact are also potential routes. As I mentioned before, DDT is still used for mosquito control in some regions, and this greatly contributes to contamination as well. Once in the environment, DDT evaporates from soil and water, becoming airborne and redepositing onto land and into water, perpetuating pollution. It can persist in soil for hundreds of years, washing into streams and lakes. In surface water, it binds to particles, settles in sediments, and is taken up by smaller organisms, starting the process of bioaccumulation. DDT significantly impacts living organisms and can lead to some very serious health and ecological consequences. As I mentioned briefly, DDT has been shown to cause adverse effects in avian species such as thinning eggshells. Populations of herring gulls, falcons, and hawks are particularly threatened by this. Additionally, elevated levels of endocrine disrupting chemicals in alligators have contributed to a detrimental decline in juvenile numbers. And in marine ecosystems, DDT is even linked to disease syndromes in certain seal species, which have resulted in population decreases. And studies conducted on mice have shown neurodevelopmental impacts which affect behavior in subjects exposed during certain reproductive stages. Most alarmingly, DDT is associated with various cancers and damage to nervous, reproductive, immune, and endocrine systems in humans, and has even been linked to birth defects. Not only does it affect organisms, but it also poses significant toxicity risks to ecosystems. Toxicity pathways include genotoxicity, oral and developmental toxicity, and disruption of hormonal systems. One study evaluated the short term toxicity in animal models, revealing a range of harmful effects, especially on the neurological, hepatic, and developmental systems. Exposed animals exhibited delayed habituation and increased motor activity. Liver damage was also observed alongside potential reproductive and developmental issues. Studies conducted on chronic oral toxicity have shown more persistent effects on hepatic function, body weight, development, and neurological outcomes. Brain development proved to be especially sensitive to exposure. In mice, doses as low as zero point 0.5 milligrams per kilograms caused impairments, while higher doses resulted in lower fetal weight, larger liver size, and tissue damage. Additionally, as toxins like DDT accumulate in the food chain, they affect species at higher trophic levels, but we'll touch on that in more detail on the next slide. Beyond its biological effects, DDT's movement and persistence shape its impact as well. Transport and sorption of the compound are strongly influenced by both physical and chemical properties. As a hydrophobic pollutant, DDT absorbs to organic matter rather than staying dissolved. It can desorb from sediments back into the water, and its lipid solubility allows it to accumulate in the tissues of organisms. This makes DDT highly bioavailable in aquatic environments where it spreads and accumulates through bodily systems. As I mentioned on the previous slide, this bioaccumulation can lead to magnification in the food chain, especially in apex predators. One study found 45 bioaccumulative DDT compounds in dolphin blubber. As apex predators, dolphins indicate ocean health and seafood contamination, making these findings really concerning. Understanding the factors that influence solubility and mobility are crucial, as these factors also affect accumulation in organisms and movement through the food chain. Solubility, mobility, and sorption are all affected by factors like organic carbon content, water temperature, and pH. Higher organic carbon reduces availability, increased temperature enhances solubility, and pH changes affect ionization and absorption. Processes like biotransformation and photolysis also shape DDT's behavior in surface waters by altering the chemical structure. Biotransformation, through metabolisms by organisms, can detoxify DDT, while photolysis breaks it down into smaller compounds using sunlight. Together, these processes affect DDT's availability, persistence, and toxicity in aquatic environments. Similar to transport and motion, bioavailability is influenced by several factors, including persistence, sorption, bioaccumulation, uptake mechanisms, and various environmental conditions. Its chemical structure makes it highly stable and resistant to degradation. Specifically, the chlorine atom in its structure enhances the stability and makes it less susceptible to breakdown. As I mentioned earlier, it's also a hydrophobic pollutant that doesn't dissolve easily. This leads it to absorb to sediments and organic matter and often reduces its exposure to degradation agents like microbial activity and sunlight. These factors and its long half life allow it to persist for decades. While studies have shown that the bioavailability in soil is declining over time, it's well established that certain fish and plants can bioaccumulate DDT and transfer it to their consumers, posing a significant threat to aquatic ecosystems and allowing it to persist. Photodegradation and hydrolysis are both non enzymatic processes that help degrade DDT and its metabolites like DDE and DDD. As mentioned before, photodegradation is driven by UV light and cleaves DDT's chemical bonds, while hydrolysis is influenced by environmental factors like temperature and pH and further transforms DDT. These processes help reduce its persistence in the environment. However, DDT metabolites can generate reactive intermediates that may damage DNA, potentially leading to genotoxic effects. While direct DNA interaction is not well documented, its metabolites can interfere with DNA synthesis, raising concerns about long term impact on genetic stability and cellular function. In aquatic organisms, cytochrome P450 enzymes play a key role in converting DDT into water soluble compounds, which aids in excretion. This process reduces toxicity and bioaccumulation. Similarly, UGTs and SALT enzymes help detoxify these metabolites. In some mosquitoes, higher levels of these enzymes help break down DDT. While this activity benefits other species, it contributes to pesticide resistance in mosquitoes, reducing DDT effectiveness and complicating malaria control. This just further highlights the need for alternative pest control strategies in developing countries. And mosquitoes aren't the only species being studied in regard to DDT exposure. A study observing fish found that DDT exposure causes neurodevelopmental issues in some individuals. Delayed habituation and increased motor activity were seen in this study and also similar ones conducted in mice. Chronic exposure led to neurodevelopmental impairments and motor dysfunction. And in the fish, DDT disrupted liver function and caused metabolic disturbances, triggering stress responses like hyperglycemia. The findings from the studies align with evidence that DDT affects liver and kidney function, further disrupting metabolic homeostasis. DDT has also been shown to suppress organ activity in fish, reducing blood cell production and altering blood composition. These changes likely contribute to reproductive and developmental issues. And beyond individual health, exposure harms aquatic ecosystems as a whole. It leads to immune suppression, gill damage, oxidative stress, and behavioral changes. These disruptions threaten the stability of aquatic environments and the organisms that depend on them for survival. Disruptions in aquatic organisms are further exacerbated by DDT's chemical properties. Its low water solubility and high hydrophobicity cause it to bind strongly to sediments and organic matter, which reduces its bioavailability. Initially, DDT degrades rapidly through volatilization, particularly in areas with high solar radiation and microbial activity. Over 50% of DDT can evaporate in such environments. Later, degradation slows and is driven more by environmental factors like leaching and sorption to soil particles, increasing its persistence in the environment. Lastly, DDT's high fat solubility is a key factor in its chemical reactivity, leading to accumulation in fatty tissues, biomagnification in the food chain, and higher concentrations in predators. The metabolism of DDT involves both enzymatic and non enzymatic processes. Enzymatic biotransformation through oxidation and reduction reactions makes DDT more water soluble and easier to excrete. Non enzymatic processes like photodegradation and hydrolysis contribute to environmental breakdown, helping reduce persistence. Cytochrome P450 enzymes not only play a role in degradation, but also in overall DDT metabolism by catalyzing oxidation and reduction reactions. These enzymes introduce functional groups that increase DDT solubility, making it easier to excrete and reducing toxicity and bioaccumulation. Additionally, GSTs are crucial for detoxification, conjugating DDT with glutathione to enhance water solubility and promote elimination. Studies show that DDT resistant mosquitoes have higher GST activity, allowing them to neutralize the pesticide more effectively. Phase 2 enzymes further detoxify DDT by enhancing solubility and facilitating excretion. These enzymes work in tandem with the oxidation reactions in phase 1 to reduce the toxic effects of DDT. Photodegradation also supports the breakdown of DDT in the environment, complementing these enzymatic pathways. Biotransformation potential is the process by which organisms convert lipophilic compounds into more water soluble metabolites through enzymatic reactions, with efficiency varying by species and environmental factors. DDT is metabolized into water soluble forms through biotransformation, primarily in two stages: phase 1, oxidation, and phase 2, conjugation. Cytochrome P450 enzymes catalyze the oxidation in phase 1, introducing functional groups that increase solubility. In phase 2, conjugation processes further enhance elimination. This biotransformation capability helps reduce DDT's toxicity, persistence, and bioaccumulation in organisms. To track DDT metabolism, scientists often analyze the chemical forms of its metabolites. This method allows researchers to follow how DDT breaks down in aquatic organisms, giving insights into its biotransformation rates. In fish, DDT primarily accumulates in the liver, where it is metabolized. The rate of metabolism varies between species, with some breaking down faster than others. Depending on environmental factors and the species, DDT's metabolites can persist from days to months to decades. Aerobic degradation, driven by microorganisms in oxygen rich environments, plays a key role in breaking down DDT. Factors like water temperature and oxygen levels influence the degradation rate. However, stressors such as oxidative damage can slow down this process. DDT accumulates in the fatty tissues of organisms, leading to biomagnification up the food chain. As smaller organisms accumulate DDT, larger predators, particularly apex predators, absorb higher concentrations through consumption. DDT disrupts endocrine, metabolic, and reproductive functions in non target species such as fish, amphibians, and invertebrates. These species are exposed through agricultural runoff, atmospheric deposition, and industrial discharge, leading to developmental and reproductive issues. DDT reduces species diversity, alters nutrient cycling, and weakens ecosystem resilience. For example, its impact on copepods, key in energy transfer in aquatic ecosystems, diminishes their ability to filter, graze, and respire, threatening overall ecosystem health. Chemical treatments and sediment removal are costly and labor intensive. Phytoremediation using plants like mustard and canola to absorb and degrade DDT offers a sustainable, cost effective alternative as plants can significantly reduce DDT concentrations in contaminated soil. The disruption of trophic balance and productivity can have profound effects on ecosystems, altering species interactions, resource availability, and overall ecological stability. Exposure in some tilapia led to incomplete axial development, abnormal cartilage formation, and reduced survival in larvae. These developmental issues were evident 10 days after hatching, indicating lasting effects on fish growth. Chronic DDT exposure caused reproductive issues in the adult tilapia and affected reproductive health and success. Tadpoles exposed to DDT showed reduced size, developmental delays, and endocrine disruptions, resulting in higher mortality rates. These effects highlight the pesticide's negative impact on amphibian growth and survival. DDT exposure leads to widespread ecological impacts, disrupting growth, development, and reproductive across various aquatic species, with long lasting effects on population health and ecosystem stability. These observed effects underscore the importance of understanding the dose response relationship to better assess the extent of DDT's impact on aquatic species and ecosystems. Many studies have found that certain DDT metabolites induce reproductive health issues in fish. Research indicates that Medaka, a small freshwater fish, is particularly vulnerable to DDT during early larval development, with chronic exposure leading to greater reproductive harm compared to exposure during sexual maturity. The pollutant DDE, a metabolite of DDT, was specifically studied for its effects on Medaka. Findings revealed that exposure to the metabolite concentrations ranging from zero point zero 0.02 to 20 parts per billion induced intersex conditions at all doses, with a 7.7% incidence at the lowest concentration. Additionally, intersex males exposed to DDE showed significantly reduced fertilization and hatching success. DDT also caused a dose-dependent increase in intersex severity and female-skewed sex ratios, with significant effects observed after just 2 weeks of exposure to 4.32 parts per billion. Prolonged exposure at lower doses resulted in even greater intersex and sex reversal effects, highlighting the numerous detrimental impacts of DDT on aquatic species and ecosystems. DDT's legacy is a stark reminder of the unintended consequences of widespread chemical use. Initially celebrated for its effectiveness in controlling malaria and boosting agricultural productivity, Its persistent environmental presence, bioaccumulation, and harmful effects on both wildlife and human health have led to significant ecological and health issues. It has caused tremendous disruption of aquatic ecosystems, reproductive issues, and neurological and metabolic damage in wildlife, including the thinning of bird eggshells and long term risks to humans, such as cancer and endocrine disruption. Although banned in many countries, DDT remains in use for malaria control in certain regions, sparking ongoing debates over its effectiveness versus its environmental and health risks. Given its alarming properties, DDT calls attention to the need for continued research, stricter regulations, and safer alternatives. It also highlights the importance of a balanced approach to public health and environmental policy, emphasizing global cooperation and responsible pest management to ensure a healthier future for both humans and ecosystems. Here are the sources that I used for this presentation.